Okay, let's dive right into this. Welcome to The Explainer. Today, we're unpacking one of the absolute most fascinating microscopic engines in the universe. I'm talking about a biological assembly line that is happening inside you right now, literally millions of times a second. We're talking about the Krebs cycle. It's the ultimate cellular factory, the one that takes the food on our plates and miraculously transforms it into the life-sustaining energy that powers every single thing we do. So here's our roadmap for this explainer. 1. The energy big picture. 2. Inside the mitochondrial matrix. 3. The 8-step cycle. And finally, 4. Vitamins, regulators, and saboteurs.
Let's kick things off with Section 1, the energy big picture. You know, it's a question we really rarely stop to ask when we're sitting down and enjoying a good meal. How exactly does a slice of pizza, or, a healthy salad, actually power our cells? We all know we need food for energy, sure, but there's this vast, incredibly complex journey between chewing your food and your muscles actually having the biological currency they need to move.
And this brilliantly illustrates that journey. No matter what you eat, whether it's lipids from fats, carbohydrates from sugars, or proteins from plants and meat, they all follow their own unique pathways of breakdown. But check out where those arrows end up. Through fatty acid oxidation, glycolysis, and amino acid catabolism, all of these completely diverse food groups are stripped down and funneled right into one universal starting molecule, acetyl-CoA. It's like the ultimate metabolic funnel.
Which brings us to Section 2, inside the mitochondrial matrix.
This engine room is host to what we call the Krebs cycle. Now, you might also know it as the TCA cycle, or the Citric Acid Cycle, which is named after the very first molecule it forms. Occurring strictly in the matrix of the mitochondrion in eukaryotes, this cycle acts as the final common pathway for the oxidation of all those carbohydrates, proteins, and lipids we just looked at. And its sole purpose? To churn out cellular energy in the form of molecules like NADH, FADH2, and ATP or GTP.
Take a close look at this microscopic geographical setting. We're deep inside the inner membrane of the mitochondria, right in the matrix. This is exactly where the citric acid cycle spins. But there's a massive catch here. Notice how the cycle connects to those Roman numerals on the membrane? Well, that's the electron transport chain. The Krebs cycle literally cannot continue without the electron transport chain working right alongside it to process the energy carriers it produces. They are just entirely dependent on each other.
All right, moving to Section 3, the 8-step biochemical assembly line.
So consider this our master map. And yeah, okay, it looks a little intimidating at first glance, I know, but we're going to break it down. Over the course of these 8 steps, the cycle just meticulously dismantles our fuel. At a high level, this factory floor process features 2 decarboxylation steps, that's where carbon is removed, 4 dehydrogenation steps, where high-energy electrons are harvested, and 1 substrate-level phosphorylation, where we get a direct, immediate burst of usable energy. Let's move to the beginning and see how this builds, starting with step 1. Our fuel molecule, acetyl-CoA, merges with a resident molecule called oxaloacetate. This whole meetup is catalyzed by a key regulatory enzyme called citrate synthase. Now, citrate synthase is tightly controlled by the body. It's actually inhibited if there's already way too much ATP, NADH, or succinyl-CoA floating around. Then in step 2, an enzyme called aconitase steps in and gently rearranges this new citrate molecule into an isomer called isocitrate. And that perfectly sets us up for step 3, which is a major event. Here, an enzyme called isocitrate dehydrogenase triggers our very first oxidative decarboxylation. Guys, this is our first huge win on the factory floor. We strip away electrons to harvest our very first energy-carrying NADH molecule. And in the process, we release our first molecule of carbon dioxide. We've officially transformed isocitrate into alpha-ketoglutarate.
But the assembly line doesn't slow down. In step 4, we meet the alpha-ketoglutarate dehydrogenase complex. This machinery drives a second decarboxylation. We extract yet another NADH molecule, adding to our growing energy treasure chest, and we release another molecule of carbon dioxide, leaving us with a brand new compound called succinyl-CoA.
Okay, let's just pause for a quick mid-cycle energy check. We're only halfway through our 8 steps, but we have already successfully harvested 2 high-energy NADH molecules, and we've successfully released 2 molecules of CO2 as waste. The factory is running at absolute peak efficiency right now, but the best is definitely yet to come.
Now, what's really interesting about this slide is the payoff we see in step 5.
Until now, we've just been collecting energy carriers for later use. But here, the enzyme succinyl-CoA synthetase performs a substrate-level phosphorylation. That means we get a direct, immediate biological currency payoff. The cycle produces a molecule of GTP or ATP right here on the spot, while transforming our molecule into succinate.
Moving right along to step 6. Here, the enzyme succinate dehydrogenase goes to work. But instead of generating NADH, it uses FAD as a coenzyme to generate FADH2. This is yet another incredibly crucial energy payload for the body, which is eventually going to be cashed in at the electron transport chain. Our molecule is now converted into fumarate.
Step 7 is swift but absolutely essential. The enzyme fumarase, which is also known as fumarate hydratase, steps in to reduce our fumarate by adding water. It perfectly hydrates it to create L-malate. And this little chemical tweak perfectly sets the stage for our grand finale.
Which brings us to step 8. The enzyme malate dehydrogenase oxidizes our L-malate. In doing so, it harvests one absolute last NADH treasure for the cell. But just as importantly, this final step regenerates oxaloacetate. If you remember all the way back from step 1, oxaloacetate is the exact molecule needed to meet the next incoming acetyl-CoA. So the loop is closed and the factory line can seamlessly begin all over again.
Let's zoom out a bit for section 4. Vitamins, regulators, and saboteurs.
So the crucial point is that this highly complex machinery requires external management, mostly in the form of your daily B vitamins, which, by the way, is exactly why your favorite neon-colored energy drinks are absolutely loaded with them. They're essential. Vitamin B1 is a required cofactor for the alpha-ketoglutarate dehydrogenase step. B2 is essential for succinate dehydrogenase. B3, or niacin, is literally a building block of our energy carrier NAD. Vitamin B5 is required to synthesize the CoA we need, and B12 is needed for the synthesis of succinyl-CoA. Basically, without these vitamins, the enzymes simply cannot do their jobs.
But just as there are life-giving cofactors, there are also dangerous chemical saboteurs. The cycle is highly vulnerable to certain inhibitors. For instance, a chemical called fluoroacetate, which is actually often used in lethal rodent poisons, strictly paralyzes the aconitase enzyme at step 2. Meanwhile, arsenite, which is derived from arsenic, violently chokes the alpha-ketoglutarate step. If just one of these enzymes is blocked by these poisons, the entire assembly line violently grinds to a halt. Energy production stops cold, and the cell dies.
Which leaves us with a truly profound realization. Considering how a single chemical inhibitor or even a simple vitamin deficiency can completely halt this delicate biological machine, just how fragile, yet absolutely brilliant, is this microscopic process keeping us alive right now? It really makes you appreciate what happens to the food on your plate long after you've taken your last bite. Keep exploring, keep questioning, and I'll see you in the next explainer.